Yeah, hello everyone. A very good afternoon and welcome to INBIOSIS webinar lecture series in this year 2021. I am Emelda Rosseleena Rohani, with today's moderator. I hope the video has provided you with some background of INBIOSIS. So for those who are interested to collaborate with us, you are most welcome. Please feel free to browse the INBIOSIS website and social media for the latest updates and information. I feel so grateful today that all of us are granted good health and time to attend our webinar series. There will be many more webinars to come, and I hope all of you will join again in the future. INBIOSIS webinar series is established to provide a knowledge and research experience sharing platform to ensure we are still connected with each other and stay motivated in our research despite this sudden pandemic. I'll now give some housekeeping rules for our webinar today. Lecture will take around 40 to 45 minutes and the Q&A session will take approximately 15 minutes, probably minimum, and probably a bit longer depending on the number of questions posted. Please submit the questions in the Cisco WebEx chat box and also FB Live page. I will read all the questions during the Q&A session. Before the seminar ends, I would like to invite all participants to switch on the camera for a photographic session. Please stay tuned until the end of the session to fill in the attendance form for e-certificate and also urus BKM credit for those participants from UKM. Without further ado, I would like to introduce our honorable speaker for today, Dr. Amit Rai from Chiba University, Japan. Allow me to share with you Dr. Amit Rai's CV. So Dr. Amit Rai got his PhD in biological science from the National University of Singapore. And he is currently the research scientist in the metabolites group in the RIKEN Center for Sustainable Resources Sciences. He is also a visiting scientist in Kazusa DNA Research Institute in Chiba, as well as also a guest lecturer in the Graduate School of Pharmaceutical Sciences from Chiba University, Japan. Without further ado, I would let us welcome Dr. Amit Rai to share his research findings. Dr. Amit Rai, the session is all yours.
Right. Thank you so much, Emelda-san. Right. So let me first set up. I hope we can go through it fine. So just a second. Let me first see. Let me first share my slides. So this is fine. Can you see my slides?
Good.
And you are able to hear me out? Okay.
Yes, very clear.
Excellent. So thank you so much, Emelda-san, for your kind introduction. My name is Amit Rai. First of all, thank you to the organizers for giving me an opportunity to talk about my research here. I would begin with a very, very quick background of my education, and then I would bring you through some of the interesting findings we have done in Chiba University and in collaboration with RIKEN. Emelda-san was, she played an important role at the beginning of this project. So that's what we were talking before this seminar is going to start. So let's begin. So I did my undergrad in India, and then I did my PhD in National University of Singapore. There I was a founding member of Graduate Student Society which allowed me to have very interesting collaborations with University of Malaya and then a few other universities. And I have visited Malaysia quite a few times. So I have a wonderful memories of Malaysia. And then after finishing my PhD, I moved to Japan in 2015 January, and since then I have been working here. My interest is on specialized metabolites. The questions that I'm interested in is plant produces diverse metabolites, and these metabolites are of very complex structure. Producing such diverse metabolite requires a lot of resources, evolution of genes, and we don't know the extent of different metabolites and then the complicated biosynthesis and regulatory elements involved. So my interest is understanding the interplay of different molecular components, and for which I take a combination of omics approaches and then try and find out the candidate genes and then the important factors that regulates the biosynthesis, and then go on to further explore across different plant species. So it's a more of a systems level understanding of biosynthesis, so specialized metabolites. Now, if we just imagine, for example, when we talk about diversity of metabolites, they are derived from relatively fewer molecules. So, for example, if in this case we see one metabolite, streptozidine, if you see this humble, simple molecule streptozidine can go in multiple directions, resulting in a huge diversity of MIA, monoindole terpene alkaloids. And these metabolites, which you are seeing here, starting from vinderline to quinine to camptothecin, they are extremely important medicine. They are proven, we are using them currently as important medicines. For example, camptothecin is an undisputably one of the most potent anti-cancer drug, and we have several medicines which are derived from camptothecin. Now, the limitation, the problem is that camptothecin cannot be synthesized chemically. So what we do, we have to rely on extraction of camptothecin from plants, and then these camptothecin then has to go through some derivatization to get you medicine. Now imagine this process, or the fact that we are dependent on plants to get the requirement of camptothecin itself makes it unsustainable. Against a worldwide demand of 3,000 kilograms, we were able to reach only 600. So there's a merit. There's a merit to understand how camptothecin is biosynthesized, what enzymes are evolved, because if we can understand that, we can then transfer the entire biosynthesis into microorganism. We can use synthetic biology approach and produce camptothecin, which we need urgently because the number of cancer patients are increasing quite significantly. Now, if we look, for example, this knowledge of, let's say, MIA, monoindole terpene alkaloids, the left-hand side figure is what we know. And this pathway is mainly derived from Catharinthus rosas, which produces Vinca type of alkaloids. And we have been able to understand and identify several of these enzymes. But camptothecin pathway, which is shown in a light yellow color box, is completely unknown. In fact, we don't know what intermediates are involved. There are some speculation in terms of some of these potential intermediates, but they are not defined yet. Now, if you look at the right-hand panel, what we know is that there's an existence of a parallel pathway. And this pathway basically includes tryptamine condensed with either sacologanine in case of Ophioryza pumila, or with sacologanic acid in case of Camptotheca acuminata, resulting in streptozidine for Ophioryza pumila, or streptozidinic acid in case of Camptotheca acuminata. Eventually, we get streptosamide, and finally, we get camptothecin. Now, one of the interesting fact is camptothecin is toxic to plants. So if a plant produces camptothecin, they need to get a very specific mutation in topoisomerase enzyme. Therefore, you can imagine plant has to first evolve the resistance against metabolite like camptothecin, and only that it can produce this compound. Therefore, this whole setup of why plants started making camptothecin, what enzymes were involved, what was the time when camptothecin started being biosynthesized, were other plants were making camptothecin, how they lost the ability of camptothecin. These are amazing, interesting questions, which if we could answer, we would be able to understand and identify the molecular components of biosynthesis pathway. And for my study, I have been working on Ophiorhiza primula. It's a plant from Rubiaceae family. It's the same family which has coffee. It's an excellent model. We have been able to have tissue culture and hair root system. We can perform transformation. So the objective of this study was to understand biosynthesis of camptothecin at the evolutionary aspect, at the genome level, at the metabolome level. And we wanted to establish Ophiorhiza primula as the model system, something like Arabidopsis or even Catharanthus roses. And the idea is that if a high quality resources and high quality knowledge is available for this species, many more people will be excited to explore biosynthesis of camptothecin, and that will accelerate the completion of this pathway. With this intention, let me first very quickly introduce you what we say when we say de novo whole genome assembly. A de novo whole genome assembly meaning that genome is not available, you have sequencing data set, and then you establish a whole genome. In the left-hand side panel, you see the starting point is starting from bottom to up. You get sequencing data sets, you establish overlaps, that results in unitics. These unitics then gets together to give you contigs. Now, these contigs are required for scaffolding that gives you a final assembly. If you see here, you have NNN. These NNN represents the assembly gaps, meaning you don't have any sequence evidence to fill the gap. Now, the main stage for any de novo assembly is formation of contigs and scaffolding. For formation of contigs, you have several technologies available. The major ones are Illumina sequencing, PacBio, and Nanopore. PacBio and Nanopore offers a longer read results, and this sequencing longer read results will basically allow you to have a better overlap. Therefore, your contigs are quite nice compared to Illumina. Now, once your contigs are ready, then you go for scaffolding. Again, for scaffolding, there are many approaches. The prominent ones are, for example, bio-nano-based. It's a optical mapping approach. And then the another one is a high-C. In case of bio-nano, the range of this scaffolding is 200 to 1 MB. And for case of high-C, library sequencing, the range is 1 to 10 MB. Now, this range is something important, and we will see why this range is so important. Now, with this, let's see about Ophiorhiza primula. So, Ophiorhiza primula, experimentally, we identified that this is a diploid genome. As you can see, A has 11 chromosomes, B has 22 chromosomes. We measured its genome size, and we identified its genome size as 410 MB. So this is the first step, and then our target was set as getting a final genome of 410 MB, 406 MB actually. Now, once we did that, we started sequencing using PacBio technology, and we acquired about 115 times genome coverage. But acquiring and running in a default mode is not always a good idea. And we spend a lot of time to optimize just the config level assembly. Now, once the config level assembly is ready, question is how you want to scaffold it. We opted for bio-nano based approach. We also opted for Hi-C based approach. But if you have config, which one should come first? Shall we use contigs assembly plus scaffold by bio-nano plus scaffold by Hi-C? Or we should use contigs plus Hi-C plus bio-nano? Or we should just use one sequencing technology, meaning we have config, scaffold by bio-nano, or scaffold by Hi-C. So, there are no clear results, there are no clear direction about it. And that's why we decided to check all the combinations. So as you can see here, the assemblers we used were Kanu, Falcon Unzip, and Falcon Phased. And we use different strategy. Like I said, the config level assembly, for example, using Kanu was scaffolded by bio-nano or scaffolded by Hi-C, or Kanu was scaffolded first with bio-nano, then this assembly got again scaffolded by Hi-C, or the reverse. So the same thing was with Falcon Unzip. And when we looked at the data, as you can see here in the table, the conclusions were, first of all, the combination, meaning using bio-nano and Hi-C together is a better approach compared to using single scaffolding technology. That's the first conclusion. The second conclusion was that the order makes sense. We checked and we found that if we are using contigs and we first use bio-nano, and then finally we do scaffolding using Hi-C, the combination is the best combination. Our contig N50 was the best. We were almost close to a reference quality genome assembly. But question is why? Why first bio-nano and then next is Hi-C is better? So if you remember in my previous slides, I show that the range of bio-nano is about 200 to 1 MB, while in case of Hi-C, the range is 1 to 10 MB. And that makes a big difference. When we use bio-nano, the config level assembly may have error. So if you see the left-hand panel, the green one is NGS assembly, and the blue ones are the bio-nano. We could identify several mistakes in assembly, which you can see shown in a red color. So from several contigs, you can see we were able to detect these consensus, the consensus were not there, meaning that using orthogonal evidence, we could say that there is a mistake in the primary contigs. So we accepted that. And why this is significant is because we are actually going in a stepwise manner. Bio-nano range is smaller resolution-wise compared to the Hi-C. Sorry, resolution of bio-nano is higher than Hi-C. Therefore, if we go in a stepwise manner of polishing, the assembly goes and results in a better way. So we checked individual contexts, we checked individual chromosomes, like in your right-hand side, you can see the small yellow color lines are individual molecules, and they show a very clear consensus supporting the assembly. Once we did that, meaning context validated with bio-nano, we perform Hi-C assembly, and as you can see here, very neat result. The gaps here is shown, for example, the blank, but you can see a very clear diagonal line suggesting that the contexts were very nicely arranged, and this is close to perfect. Now, what here we have done so far, we have first optimized primary assembly, validated using bio-nano, then we again validated by Hi-C. So you can imagine we tried all possible combinations. So what we would like to believe? We would like to believe that the assembly is absolutely correct. The answer is no, assembly is not absolutely correct. So when we finalized our assembly, we got only 21 assembly gaps. So gaps are, you have two, I don't know if you can see, you have two scaffolds, you have two contexts. And what you do is you have no sequencing evidence in between. So what you do, you connect them and you put them as a gap. So we asked if the gaps, the orientation is correct, because for gaps you have no sequencing evidence. Therefore, it could be this way or it could be the other way. So we checked that. So in this plot, you see in the left-hand side, you have a blue bar. In this blue bar, you have one gap. You can see assembly gap here. And based on our initial assembly, we designed primers as shown here, 79648, and so on. So ideally, 796 was supposed to be far from the gap, and 48002 was supposed to be close to the gap. But when we checked through fish analysis, we found the reverse, meaning 48002 was far from the assembly gap, the centromere. What this means? This means that even if we use multiple sequencing and scaffolding technology, the gaps are the places where we are supposed to see error. And that's why experimental evidence is essential if we are talking about a high quality genome assembly. We did that actually. We validated each and every gap. We took representative sequences and we looked at their position across the gap. And for example, if let's say in chromosome number 1, we expected this the signal to be far away from the gap, and we were able to see that. Now, if the signal was flipped here, we would have believed that there was a mistake, and we would have flipped the context. So what this means? This means Ophioryza pumila is among very few experimentally validated genome assembly reported till date. People don't give, they really don't care about this fact, but then this is very important because all the downstream analysis is going to be affected if you have not done the experimental validations. So the approach that I'm explaining here is we started with PacBio, tested different assemblers. Once our contig level assembly was ready, we validated using BioNano. Then after the conflict resolution, we used Hi-C. And as you can see here, our assembly was already quite high quality, but then we further validated experimentally, again using BioNano dataset. We checked the error, we filled the gap, and finally, genome assembly of Ophioryza pumila became with a contig N50 of 18.5, very rare. Not many plants have that, and gaps of only 21. The only plant which has such a few number of gaps is maize or rice. So again, the point is among medicinal plants, this is the best plant genome assembled ever. Of course, again, we use the repeat sequences, and then we could localize the centromere, as you can show here, as you can see here in the right-hand side. So what you see here, the genome features in your right-hand side, where we have shown from different repeats, assembly gaps, repeat content, predicted genes, and so on. And in the left-hand side is your genome features. In the right-hand side, you can see Ophioryza pumila genome assembly showed 97.1% completeness, and we were able to phase Ophioryza pumila into haplotype 1 and haplotype 2. So again, Ophioryza pumila is a phased genome where we have haplotypes information as well as the reference genome. And in terms of BUSCO score, is one of the most complete genome assembly ever reported. When we place Ophioryza pumila compared to the other plant genome reported and published till date, as you can see here, Ophioryza pumila is among the best with only 21 assembly gaps. The other red color circles are representing plant species which produces MIA, monoindole terpene alkaloids. So again, this is just trying to show that Ophioryza pumila genome assembly therefore represents the most complete genome assembly and is suitable for further investigation of MIA biosynthesis. Now, once the genome assembly is ready, what we do, we want to start exploring its features. We want to know how similar or different this plant is with respect to fellow species. So if you see in the left-hand side panel, the first figure is the content of LTR. Now, Ophioryza primula entire genome consists of over 50% of its genome is of repeat. Compared to that, Chemtotheca acuminata, another chemtothecin producing plant, showed about 30% of genome was repeat. So again, suggesting that the repeat content of chemtothecin producing plant Ophioryza primula and Chemtotheca acuminata is very different. Not only that, if you notice, the dominating repeats of Ophioryza primula was, for LTR element, was gypsy. Compared to that, for Chemtotheca acuminata, it was just opposite. In case of Chemtotheca acuminata, it was copia. When we looked at KS distribution plot, we could find that in case of Ophioryza primula, there was a single peak. And then a representative suggesting whole genome triplication happened for Ophioryza primula, but after that, no whole genome duplication happened. Compared to that, we could detect in case of Chemtotheca acuminata that we found 2 peaks in case of acuminata, suggesting that Chemtotheca acuminata undergo recent whole genome duplication. Now, again, this is interesting because this suggests in case of Ophioryza primula, no whole genome duplication occurred. In case of Chemtotheca acuminata, another chemtothecin producing plant had whole genome duplication, and whole genome duplication influences a lot in terms of evolution of new enzyme, biosynthesis of specialized metabolites. So therefore, it suggests that probably the evolution of chemtothecin biosynthesis happened in a parallel way. In fact, when we measured the evolution of the timing of emergence of these plant species, we found that Chemtotheca acuminata, which we reported recent whole genome duplication, this was an ancient plant, and this got speciated about 120 million years ago. Compared to that, the coffee and Ophioryza primula got separated about 47 million years ago. The MIA producing plants branch, which is here, was actually evolved about 68 million years ago, where it got speciated into Ophioryza primula, the chemtothecin producing plant, and Catharinthus rosis and other MIA producing plant about 58 million years ago. So in conclusion, what we can say for sure is, at least in case of chemtothecin producing plant, the type of repeat content, the expansion strategy, which means in case of Ophioryza primula, no whole genome duplication. In case of Chemtotheca acuminata, whole genome duplication happened. And in terms of evolution timing, clearly chemtothecin biosynthesis took a parallel direction of evolution, which is also consistent with the biosynthesis pathway I showed to you just before I started this presentation. Now, because Ophioryza primula belongs to Rubiaceae family, which has coffee, of course, we compared Ophioryza primula genome with coffee. And you can see, except chromosome number 2, it's absolutely collinear from chromosome 1 to chromosome number 11. Now, for chromosome 2, you can see coffee chromosome number 2 has 2 contacts. A part is attached or showing a linkage with chromosome number 9 of Ophioryza primula, and the remaining part is showing contact with chromosome number 2. Now, you see here, if we look at the gaps, for coffee canifera, it has thousands of gaps. But in case of Ophioryza primula, it has only 21 assembly gaps. So this could be biological relevant, meaning that indeed such relationship exists, or maybe this is assembly error. And we showed, I'm not showing it here, but we showed using coffee canifora, we suggested that maybe this is a mistake. Maybe this is an error in case of coffee genome. So again, the point here is that experimentally validated genome assembly could serve as an important reference to check the quality of other genomes. And again, this is a learning for all of us to understand the importance of experimental evidence for a genome assembly. Now, I think, I hope that till this point, I was able to convince you the method we use to achieve this genome assembly, and to convince you that Ophiorhiza primula is indeed one of the best plant genome. But in terms of metabolome, like I said, plant produces thousands and thousands of metabolites, but we don't know actually. We know the main guy, we know the main metabolite, but we don't know all of them. So another objective for this study was to understand what metabolites are produced in Ophiorhiza primula. And for that, we developed a method which was published in Nature Method, where what we did was we performed a complete isotope labeling of metabolome. So for example, in case of Ophiorhiza primula, we cultured Ophiorhiza primula in natural media, the MS media, and let's say, and then in another case, we grew Ophiorhiza primula hay root in a media which has complete replaced carbon source with isotope labeled 13C. And the difference here is, as you can see here in one, that you can identify metabolites which have got isotope labeled, and that gives you a confidence in terms of identifying real metabolite features. Using this approach that we published, we reported about 4,000 metabolite features of Ophiorhiza primula. And we further expanded this because our focus was on monoindole MIA. And therefore, what we did was we performed complete nitrogen labeling approach. Using same approach, using same strategy, we discovered over 200 metabolites in case of Ophiorhiza primula. The panel, the left panel you see here, these represents a comparison of Ophiorhiza primula metabolome that we discovered with other plant species. And what you can see here is that overall, Ophiorhiza primula was quite distinct in terms of identified MIA, which was almost absent in other plant species. Not only that, please look at the red circle. It's actually keep on going very nicely concentric. What this shows is that the specialized metabolites for these individual plants are the signature. So the specialized metabolite we discovered for Ophiorhiza primula was nearly unique for Ophiorhiza primula, and the same thing was true for other plant species. When we identified these metabolites using isotope labeling approach, the accumulation pattern showed that majority of MIA shown in black dots are highly accumulated in the hay roots and root tissue, while the level was significantly low in case of leaf. And this is consistent with what we know, that the hay root and root tissues are known to accumulate MIA and camptothecin, while the leaf tissue has less level of these metabolites. We then performed transcriptome profiling for the same tissues, and we performed metabolite gene network analysis. So as you can see here, the brown color line dots are the genes. We looked at their co-expression with these metabolites, and we used gene co-expression and gene to metabolite co-expression analysis to assign putative genes to the biosynthesis pathway, which you can see here. Now, if you look at the left-hand side panel, this is a sacchoiridoid biosynthesis pathway, which results in the key metabolite sacchologanine, and tryptamine comes from tryptophan. And these two gets together to give streptozidine. Now, As you can see here, the biosynthesis of these metabolites were specific to hay root and root, and the expression of genes were consistent with metabolites. They were highly accumulated, the expression was highest in the hay root and root. So metabolites and genes are correlated. Now, if you see here, again, the same thing was true for the known, the homologs of known enzymes involved in MIA biosynthesis, as you can see here in the right panel. What's important here is these black dots. These dots represent the assignment of these enzymes to the metabolic gene cluster. So the point I'm trying to make here is that the genes which we assigned to MIA biosynthesis were highly correlated with metabolites we discovered. And not only that, all these enzymes were part of a gene cluster, suggesting that gene cluster probably are the key places where we can mine and discover new enzymes. We therefore looked at, for example, these enzymes, how were they evolved when we compared, for example, Ophioryza pumila with the other producing plants. We could identify enzymes which were specific, and we could identify enzymes which are conserved across the MIA producing plant, and orthogene families specific to MIA producing plant. And why this is important? Because you would like to believe that if Ophioryza pumila or other MIA producing plants are different from other plant species, they need to evolve some enzymes which help them to direct the evolution of specialized metabolite in a specific direction so that they can produce a different class of metabolites. So what here you see is the orthogene families at the bottom, and the red lines are basically those enzymes, the one we are seeing through this arrow. These are those enzymes which were specifically expanded in MIA producing plant. And this is very important because, for example, this STR is the step which synthesizes the strictocidine. If the strictocidine biosynthesis occurred, then the chances of native enzymes to direct it to MIA biosynthesis increases. Therefore, if a gene family is expanding in the biosynthesis in the plants which produces MIA, of course, we would like to believe that that probably played an important role. So several of these key enzymes shown in the red lines are the ones which were specifically expanded in MIA producing plants. Most significantly, it was SLS and STR. Here, for example, in case of STR, you see, we discovered two orthogenes, and they were very nicely grouped into two parts. The part which is functionally characterized STR was specific to only MIA producing plants. And this, when we performed busted analysis, we identified that these genes were positively selected, meaning a plants require to select this plant and this enzyme and keep it if it wants to retain the biosynthesis of MIA and this chemotype. Now, when we looked at the KS distribution analysis, so you see here in the left-hand panel, KS median, if KS value is bigger than 2, let's say, so 2 is regarded as the point when whole genome triplication happened. The greater the value, the older the gene. The smaller the value, the newer the gene. The red color represents the point where this STR 15245 gene family evolved. As you can see here, the number of yellow dots are significantly higher in case of when we draw a line here through STR. And these yellow dots are representing MIA producing plants. So what left-hand panel suggests is that the evolution of enzymes that are involved in MIA biosynthesis occurred recently. It occurred after STR was evolved. Not only that, when we looked at, because we discovered that the biosynthesis of camptothecin is a parallel, evolution was parallel. So we looked at, for example, in case of Camptotheca acuminata, whole genome duplication point, you can see that majority of these enzymes, which are putative MIA producing plants, actually already evolved before whole genome duplication of Camptotheca acuminata, suggesting that although the genome got expanded in case of acuminata, probably the majority of enzymes which shapes the evolution of MIA already occurred before this whole genome duplication event. Meaning that although of Camptotheca acuminata got spatiated about 120 million years ago, the content was conserved and that retained its biosynthesis pathway. But this also means that some plants lost it. How they lost it? So we looked at, for example, gene clusters. So as I just mentioned, we looked at, so this in the right-hand panel, what you are seeing is chromosome. And this is chromosome number 2. And what you see here is we reported 6 gene clusters. And in this case, these are the orthogene families. And we could show that several of these gene clusters were conserved across producing plants. MIA producing plants is Ophiorhiza pumila, Camptotheca acuminata, Catharethis roses, and Gesremium. We could show that the enzymes we assigned to MIA biosynthesis, the gene clusters are actually conserved in these genes. And you have to remember that other than Camptotheca acuminata, other than Ophiorhiza pumila, other plant genomes are crazy fragmented. Therefore, you would like to believe that the evolution of specialized metabolites was centered and protected along the gene clusters. Let me show you very, very interesting example. So if you remember, I explained to you that a cycloidoid biosynthesis pathway gives you cyclogamine, and then tryptophan gives you tryptamine, which gets together to give strictosidine. The two enzymes, TDC and STR, is essential to produce this strictosidine. And if you have a strictosidine, then only you can get MIA plants. When we looked at and when we scanned the gene clusters, we found that the TDC, functional TDC, and functional STR are conserved as a gene cluster. So Gelsemium, Sempervirens, Catharethis roses, and Ophiorhiza pumila, they were conserved in the order. Now, if you remember, I said when we compared Ophiorhiza pumila with coffee, I said they were brothers. But what's very interesting thing was that coffee lost one enzyme. What was that enzyme? That enzyme was STR. And what is this STR? STR is the enzyme which first evolved, and that allowed evolution of other enzymes, which then eventually leads to MIA biosynthesis. So what this suggests is that probably gene clusters are the sites where the important enzymes are retained over generation, over speciation. And if that key enzyme is retained, the chemotype is conserved. Therefore, gene clusters are potential sites to discover biosynthetic enzymes. And probably it's better to look for those gene clusters which are conserved across plant species with similar chemotypes. So here I'm just showing, I'm almost done. So here I'm showing one example. This is on chromosome number 5. These are 3 gene clusters I'm trying to show you here. And the dots are basically the individual enzymes you are able to see. Now, for example, let's say we look at this first plot. What this shows is the gain or loss of this enzyme across different plant species. You can see that this enzyme was specifically expanded in Ophiorhiza pumila and was retained in other MIA producing plants, but was absolutely absent in other plant species. This suggests that this enzyme, retaining this enzyme was essential and was useful for the evolution of novel metabolites or specific chemotypes. So effectively, this means that gene clusters, especially those which are conserved in plants producing similar type of metabolites, are important to discover, to investigate, and to do functional characterization. And if they are co-expressed, correlated with the type of metabolites that we expect they are producing, then these are the genes we need to go further and perform functional characterization. So in summary, I'm almost done in 45 minutes. So in summary, we showed in this study that we published this year in Nature Communication that hybrid genome assembly is the strategy to use for high quality reference level genome assembly. Why? Because the stepwise polishing genome assembly is better. It helps us to reduce assembly errors at different stages. And if we can reduce these assembly stages, we will be able to get a very high quality assembly. Another learning from the study was that despite using a hybrid approach, the assembly gaps are the places where you can still find the errors because you have no evidence, you have no sequencing evidences there. Therefore, experimental validation is essential. If you can't do that, then you should not really confidently say that these gene clusters are there important, because gene clusters are the order of genes you have it there. So through this study, we established a gold standard reference and phase genome assembly of Ophryza primula. And we also used isotope labeling approach to discover over 273 plus nitrogen containing metabolites. Many of them, or majority of them, are MIAs and probably intermediates of camptothecin biosynthesis. So this resource is very important for us to investigate and discover some of these intermediates that are not known, because in case of camptothecin biosynthesis, we even don't know what are the intermediates there. Comparative genome analysis we used, and we were able to identify the key event, which was evolution of STR. STR emergence allowed biosynthesis of streptozidine, which then through a random biological processes eventually resulted in evolution of MIAs. And if STR was lost, for example, in case of coffee, the evolution of MIA stopped. Therefore, the discovery of enzymes which are of importance in these plant species, gene clusters are the important site. And we also showed that gene to metabolite network together with metabolic gene clusters are the way forward if we want to really discover and complete biosynthesis of our metabolite of interest. With this, I would like to acknowledge this is currently my team at Retain. All of them are brilliant scientists. We got a lot of suggestion and collaboration across Chiba University, Katsa DNA and Research Institute, and CSRS Retain. The metabolome analysis and experiment was done at Retain. The genome assembly and analysis was done at Chiba University. Sequencing was done at Katsa. We were very generously supported by Kakenhi and Chiba University Institute of Global Prominent Research. With this, I would end saying that, yeah, we need to establish sustainable resources. There is a very urgent need. If we completely rely on unsustainable resources, the world is going to end. So we have to put our effort and then find these solutions which could help us to, establish resources that our next generation could use to survive. With this, thank you so much. I would be very happy to take any question. Right.
Thank you very much, Dr. Amit Rai. It's very interesting study and a lot of knowledge that you shared with us. So without further ado, I will read the questions from the chat box. The first one is from ago from life, he was asking why is the Illumina reads were not used to correct the PacBio reads from the beginning.
Well, there are initially when PacBio sequencing started, the amount of errors in PacBio was quite high, and that's why Illumina polishing PacBio reads polishing by Illumina was essential. But now, when the sequencing technology and the new chemistry evolved, the amount of error in PacBio reads are not that much, and these errors are random. Therefore, the overlap-based assembly by itself corrects these errors. So before the assembly, the modules are first overlap-based or MHAP-based read correction, then trimming, and then assembly. So actually, it's better not to correct error beginning, because eventually, once your assembly is ready, you will polish your assembly using Illumina. So if you see my this step, after assembly, final assembly, we did validated, we did corrected assembly using Illumina. But I think it's better not to use Illumina at the beginning, but probably at the end to completely polish and improve your genome assembly. So I would not do it, but yes, I think 3-4 years back, yes, that's the way it was.
Chromosome
Emelda,
number
I can't
assigned
hear you.
arbitrarily or based. Was the chromosome number assigned arbitrarily or based on previous studies?
Chromosome numbers were assigned based on synteny analysis between coffee and Ophiorhiza pumila. So because both of them are from Rubiaceae family, we did a synteny analysis and we found that they were super collinear. So if you see this figure here, chromosome 1 of coffee canifora showed very high collinearity with this chromosome. Therefore, we called this as chromosome number 1, and so on. So numbers were based on coffee canifora. And yeah, I mean, you can actually do it arbitrarily. Few people rank them based on the chromosome size. The bigger gets chromosome number 1, the smaller gets last chromosome. So yeah, I mean, it's up to you. In our case, we used coffee canifora to number them.
Okay, thank you. In this study, both Coffea canifora and Ophiorhiza pumila has the same chromosome number, as you said before. The collinearity was absent in one of the chromosomes. So could you please explain further, showing the evidence on how this may have been attributed by the gene error?
Actually, what then we did was, so for that you should read this paper. But what we then realized in case of Ophiorhiza pumila, we had only 21 assembly gaps, and we still found one error in chromosome number 2. Compared to that, in case of coffee, we have about 7,000 assembly gaps. So it's impossible to check everything. But what we then did was we created a probe for checking. Instead of using coffee canifora, we use coffee arabica, which is easier for us to get that time. And coffee arabica is a hybrid of coffee canifora and another coffee, I am forgetting the name. And we hoped that, for example, we created a probe here, this side, which is contact with chromosome number 9, and another probe on this side, which is in contact with chromosome of Ophiorhiza pumila, chromosome 2. And we hoped that if there is an assembly error, these probes should be on 2 chromosomes in case of coffee arabica, because again, we believe that coffee arabica has coffee canifora part. And we showed, I think it's in the supplementary figure, we showed that indeed the probes are 2 different chromosomes. So this led us to believe, or rather propose that probably coffee canifora has assembly error. But to be very fair, to be honest, this experiment should have been done on coffee canifera. That has not been done. So at this moment, I'm collaborating with another professor at Chiba University, and now we are doing this experiment on coffee canifera to see if this is an assembly error. If this is an assembly error, it's a very end of the story. But if this is not an assembly error, that's a very exciting news for me, because that means this could have some biological relevance. So we are now exploring in coffee canifora now. We have got the plant, and hopefully we will know that if indeed it's a error that we proposed, or if it's real. I mean, this relationship is real. Yeah, that's ongoing now.
What do you mean by the biological effect?
So because
on the
yes,
error?
because for example, so if there is error, then end of the story, because yeah, it's an error. We can't say anything. But imagine if it's not an error. If it's not an error, meaning that chromosome 2 has contact a massive chunk. If you see, it's not a small part actually. A big part of chromosome 2 actually has a relationship with chromosome 9 of Ophioryza pumila, and this suggests that, because you know Ophioryza pumila and coffee canifora evolved around 40 million years ago. They got speciated, and although they are supposed to be very collinear, the difference or the mixing, if you could say, happened in between chromosome 2 of coffee and chromosome 2 and 9 of Ophioryza pumila. So then the question would be, if this happened, what changes it could bring? What genes gonna change? Does it have any influence in terms of the transposable elements comes up? So that then starts a very interesting, very different question. If this is real, what could have happened? How it would have helped to shape the metabotype of coffee and the metabotype of Ophioryza pumila? So those are the questions. So we are testing now. If this is an assembly error, we are done. But if this is not an assembly error, then you know the question increases, and that becomes more exciting for us.
Thank you very much. Yeah. Okay. Let's go. Let's move to the next questions. Okay. So what is the current status of identifying the missing enzymes for the targeted compound?
We have started experiments. There are different stages. So, like I said, what we have realized is mostly we use. They are on different experiments are going on. We have few positive guys.
Yeah.
We will publish them soon. Right.
All right. Yeah. On the biosynthetic pathway, is it?
Yes.
So you solve
like
all
we.
the.
No. That would get me a Nobel Prize. No. We have done. We have established metabolome data sets, and we did a very interesting experiment, which I would not say what we did, but then that allowed us to discover several intermediates, several new intermediates that has not been reported before. And what then we are trying to do is we are trying to use chemistry to assign them the position where it could stay from streptozidine to camptothecin. And that's actually ongoing. We are also trying to see if these metabolites are localized. So we are trying to do, we are trying to do imaging mass spec. So that's actually ongoing. And like I said, we performed gene cluster analysis together with co-expression and correlation between genes and metabolites to discover several candidate genes. And based on the enzyme types, you can always predict what could be there. And those functional characterizations are also going on in parallel. So I think in terms of future results that we hope to see soon would be probably identification of some of these intermediates, placing them in putative pathway, and probably some of these enzymes. So we hope to make Ophioryza pumila like catecholamines, probably so.
Wow, that is very interesting. So we will wait for a paper, which is maybe we publish soon now.
I hope so. I hope so. Right.
Okay. Wait. Let me search for the next question. Okay. In this time, comparing between Coffea carnivora and also Ophioryza pumila. Do you have any current study using other plant species, same family of Rubiaceae?
No. So I'm not. Yes. Actually, again, it's a secret. I can't tell, unfortunately. But we have a plant in Rubiaceae family, which is very close to Ophioryza pumila, but it doesn't produces camptothecin. But it does produces some of these MIA guys. And what I have done is I have sequenced its genome, and scaffolding is ongoing. I have also sequenced genome which produces camptothecin, but it's very far. It's not from the same family, but it's a very far distant family in the producing plants. And the idea here is to understand, using these producers and non-producers, I want to really discover some of these genes. Now, few of you would say, but you already have it. Actually, the quality of genome assemblies we have is not good. The new sequencing data set has already been generated, and I'm now assembling them. Hopefully, that would give us many things. It's very, very exciting. I can't tell, unfortunately, but one of the plant genome is super massive genome, and I think that the expansion was because of the transposable elements, and therefore it would be very interesting to see the role of whole genome duplication and transposable elements for the evolution of camptothecin. It's a completely different dimension we are looking at. So that would get us many, many things. So that's ongoing. Hopefully next year. Hopefully.
Yeah, we wish you the best. So we will wait for a paper to publish for next year. Okay, there's a continuous question here. Do you think it is doable to missing gene or metabolites in the pathway of interest based on the metabolomics and transcriptomic data only? This is due to because we have any genome information of the plant of the interest.
Well, it's a bit tricky.
Do
You
we get
see,
the
when you
question?
have only yes. I think let's say if you have no genome, you can always have transcriptome sequencing you can do, and then you can establish de novo transcriptome assembly. So yeah, that's possible. Once you create de novo transcriptome assembly, then you can do expression analysis. You can then use metabolome, and then you can combine metabolome and transcriptome to have correlated metabolites with co-expressed genes and so on. The challenge here would be, in most cases, the strong candidate genes would not be complete. They will be fragmented. So I think Emelda-san knows better. You have to do race experiments so much to get the full length for experiments. There are other challenges, but like, only recently the whole genome sequencing has become more accessible. Before it was not there, but the discovery of new enzymes was continued. So it's not impossible for sure, but it's definitely challenging. You will have more candidate genes, you will have to find full length. If a closer species has its genome published, it's better. you can then use those sequences and try and guess what's there and so on. So it's possible, but tricky for sure, difficult for sure.
Okay, thank you. So this is from Dr. Ng Chiang Leong. Thank you for the interesting and insightful talk. Streptococcus synthase gene that missing in the Coffea carnivora shows very good example of chemotype specificity of plant. As STR or streptococcus synthase, streptococcus is located quite upstream in the MIA pathway. I wonder similar study approach suitable finding more specific enzymes that involved in particular compound of MIA, which is at the more downstream pathway.
So why streptozidine attracted our attention, and the way we have described it was because when we did the evolutionary analysis, we could see that STR was an old enzyme. It was not a new enzyme. And what struck us quite interestingly was that the STR enzyme is present across all plant species, not only Ophioida pumila. But then these STRs evolved into specific enzyme which was retained. Now, coming back to your question, actually, we could find gene clusters which were conserved across MIA producing plants, and specific genes were missing in coffee. We have those gene clusters, we have seen them, but because these genes are not characterized, we don't know. Maybe some of those genes are downstream genes, we don't know. So that would require functional characterization for sure. Some of these are, we are already started doing some of their functional characterization, but we also need to remember that my hypothesis of evolution and diversification of specialized metabolite is creation or biosynthesis of one metabolite, for example, streptozidine in this case, was important. Now, plants has thousands and thousands of genes. Many of them has multiple specificity. And effectively, those enzymes then could take these substrate to produce a variety of intermediates, and some of them could provide benefits. That would result in positive selection of those genes. So for me, like your question, missing is important, but at the same time, the localized duplication is equally important, because along the chromosome, if one gene gets localized duplicated and that got specialized, that evolved a new function, most likely that's the guy we should be looking at. So we have discovered some of those which are missing. We have also discovered some of them which have got duplicated, and probably they are the potential candidates. So we have those. We do think that they are downstream. We are working on that. And the same principle applies to the other species, other chemotypes. So yeah, we are doing that. I agree with you. Yeah, I understand your question. I think we are in that direction too. We believe that should work. Right.
Okay. Yeah. All right. So this is from Ching Ching. With the recent improvement in technology, we can obtain high accuracy assembly once read coverage is high. Do you think Illumina read is still a must to do error correction for this project?
If I do this project today, I will not do it, because now you have high fidelity packed biosequencing. That is almost Sangus level, 99.99% accurate. Then you really don't need polishing by Illumina. In fact, even PacBio-based, overlap-based error correction, it brings you close to 99%. But then, you want to be very, very, very accurate. But after this hi-fi, I think it's not needed. unless you want to look, for example, a very minute SNPs and so on. Yeah, it's maybe important, but otherwise for assembly polishing, you don't need anymore.
So it depends on our objective, on what we are searching on. Okay. So thank you very much from them, from those who are asking, Dr. Ng Kianya, and also from Chin Chin. Before was questions from Dr. Sharuna Takaim Baharum and also Dr. Go. Okay. Is there any questions? Okay, looks like everyone is okay. Yeah, we have no more questions anymore. But so I think it's already 4 o'clock. So we will move on to our next session. So let me check again. All right. Okay, we are done with all the questions. Okay. So we have come to the end of the webinar. Thank you very much, Dr. Amit Rai. Oh, actually, I call him Amit Sensei before because he is my co-supervisor. So thank you very much, Dr. Amit Rai, for the very interesting subject. And I'm sure has opened up our view on and give an idea on how we want to bring our research output to the next level. So today's webinar had recorded 83 participants from the Cisco WebEx and also 20 participants on the Live page, the INBIOSIS UKM. I also would like to thank all participants for spending your time with us today. Please stay safe and take very good care of yourself, okay, because of this pandemic currently. Before we say goodbye, let us immortalize this session by switching on video and have our face captured. So it's like a photography session, okay? So let us on our video, everyone. I'm invited to on your video. Okay, please wait a minute. We will wait for more to come in. Okay, Intan standby.
Okay. You're still waiting. Okay, ready?
Okay, I think.
Okay, 1, 2. Alright. Okay, one more. Okay, 1, 2, 3. Okay, alright. Thank you.
Okay, Amit Sensei, please don't leave the session yet. We will have the session later. Intan. Okay, so thank you very much, Dr. Amit Rai, for the very fruitful and insightful talk. So thank you, everyone. Please stay safe and very good care, and also stay tuned to our social media for the next webinar. For the next webinar in INBIOSIS. So thank you very much. Bye.